Now we're glad to have this panel discussion, and I really want to thank Raphael for taking the lead on this. Appreciate that very much that he's doing this, and I want to give it over to you.
Well, thank you very much, Marcus, and I would like to welcome everybody for zooming in to this roundtable discussion as part of the 5th Gateway NMR conference organized by Vanderbilt University. My name is Raphael Buschweiler, and I'm at Ohio State. I would like to thank the organizers first to move this meeting to the virtual format and accepting the extra work that went along with this step. But I really believe it's important that we continue to discuss science and also issues surrounding NMRs and facilities in our community, whether it's in person or whether it's virtual as today. So the title of this roundtable discussion is NMR Challenges and Opportunities During and Post-COVID-19. And we all know this coronavirus, this novel coronavirus, it came up on us early this year with very little warning, and it had a profound effect on all aspects of our lives, including the way we do NMR research now and also in the foreseeable future. And unfortunately, we're not out of the woods yet, but at least we all got some experience how to deal with this new and still very risky situation. Therefore, today we have 2 expert panels. The first panel will discuss questions and challenges surrounding shared NMR facilities during and post-COVID. Most participants at this conference are users of such NMR facilities, but most of these facilities, or probably all of them, have not been designed with a pandemic such as this one in mind. Therefore, with this first panel, we would like to discuss and share experiences and challenges along with measures that the panelists and their universities have taken in response to COVID, want to learn how they have played out so far. The hope is that we can learn from each other's experience to provide a safe and effective research and training environment in our NMR facilities. So let me introduce our panelists. I'm really grateful that they all agreed to participate today. We have Jasna Veja at University of Massachusetts Amherst, Johannes Leisen at Georgia Tech, Minli Jing, University of Michigan, and Alex Hansen at the Ohio State University, and Marcus Furler, whom you already know, at Vanderbilt. So the first question I would like to ask, it would go to Alex Hansen, is how is your experience? How was your experience to operate the shared facility during COVID?
Yeah. You'll stop other screen share. Can I share screen?
Yes, absolutely.
That one. So the experience has been quite interesting, I would say. In general, I mean, at the beginning of the year, everything was just shut down. I think we were pretty quick to start shutting things down even before the university declared it, to prevent people from coming in, and starting to strategize our plan of how to work with users. And then for several months, there's just nothing. But by June, there was definitely a big demand to be running experiments again. And so we had to come up with ways to mitigate that, right? So we have the 2 facilities at OSU. And the first and foremost thing we decided was we need to have like a single flow of users through the lab. We can't have people congregating or crossing paths and things like that. And so and the first priority entering the lab became you have to wash your hands. And so we set up various directions in order to get people to the sink, and we provide lots of soap and hand sanitizer for the users, space to leave their samples while they're doing that. And then another big key part was, I mean, especially in the Rife facility, we haven't had, I mean, it's a little bit more of a tight spacing. And so we had to come up with these barriers in between instruments, instrument consoles, because it's very difficult to maintain the 6-foot spacing. Whereas in CBEC, there's a lot more space. And so we were able to sort of, we have rules in the, from the facility to allow a certain amount of square footage per user. And so we just sort of made a very clear indicator of where people should not cross if somebody's doing work. And then, of course, we had to sort of come up with the rules and for how people are supposed to behave in the lab. And we ended up, I mean, we do a lot, we encourage, well, we require this users to wear their masks while they're doing work. They clean up the workstation before and after using 70% ethanol. And we actually use these sort of press and seal or saran wrap to put over the keyboard while you're doing work. And we opted for that as opposed to requiring gloves because I mean, being in a chemistry lab, we just sort of were of the opinion that, or we didn't want to encourage people to be wandering around the building with gloves on, right? And that would completely defeat the purpose. So this still requires that there's a constant cleanliness of the workstation and that sort of thing. And then, especially around June, the main real helpful piece was that we have all of these sample jets, especially for the chemistry instruments. And this has been key to, I mean, as soon as some researchers were allowed to be back on campus, the chemistry side of things like quickly hit capacity once again. But at present, we are running all of those samples. And so we have a nice organized way to drop off samples where people don't have to interact, and our staff will pick up the samples and just put them in according to the number of on the drop off, and the users can queue up Icon NMR remotely and run things like that. For the biomolecular side, we quickly got together to update all of our spectrometers to the latest CentOS 7 and TopSpin 3.6, and enabled a lot of useful remote access. And so with sort of easy handoff of samples and the remote access and the automation, we've been able to really quickly come back to normal on some levels. The main downside is just some inconvenience of having to do all of the sample running ourselves, as opposed to how we would prefer to have users running things.
Thank you, Alex. Jasna, how was your experience at the University of Massachusetts?
It was similar, but we were open mostly all the time, except at the very beginning when we were all caught by surprise. So I can share my screen as well.
Yes.
We have a very different facility than Alex's. So we have 5 instruments in 4 different labs. And so then there is definitely much more privacy. You really don't need to follow how people come and how they leave. What we needed to do was to come up with the guidelines. And since we don't have sinks in our NMR labs, so we had to do heavy alcohol, everything, and gloves. So this is the guidelines that we have used for each lab. And so what is happening, you know, of course, students, postdocs, users, when they come, they sanitize the working surface, and their gloves needed used to only add a spectrometer, and then we have benches for chemistry work, but that we have limited, so people should come with samples already in NMR tubes. And so we have then gloves on used all the time, but separate gloves if somebody has to transfer a sample from Eppendorf tubes to NMR tubes. We even had outside users, meaning people from Boston area, from a biotech school, came during the major lockdown because university policy was that you have to have a plan of continuity research of the research, and once when it is accepted, then you are cleared and then you could be coming. We could not have new users, so only those experienced users, and unfortunately, this still holds, no users in the facility at all. So what I have done during the summer, I had some students that I trained online for a few months and then managed to sneak them in September when things kind of, when the undergrads are actually allowed at university. So basically now for us, it is still a problem how to work with students in person because not that many facilities at the university, I mean, we have 30, are very happy of having to work in person. So as you can see, we have KN95 masks and shields, and we try to keep distance of close to 2 meters. And this is, I do this with students who I have worked with before, so these are not novices, but everybody needs a little bit enforcement from time to time. So this is still done on a small, in limited cases because you know, you have to be careful. One thing that really helped and made a major breakthrough was when everybody who is in the lab in the university started being tested twice a week for COVID. So at least, you know, I would schedule people that I have to work with like a day when they receive the test results, and then we would work that day, set up everything, and then after second testing, we would meet again. So that's our story. And so we have also worked as everybody in various phases, but right now the policy is following that we still cannot have more than one person at the instrument. In the room within the lab with 2 spectrometers, we can have 3 people. One of those 3 will be instructor and only 2 students. So this is what we are trying to keep. And as you can see, I just listed few facilities here. Nobody at university is allowing new users, and I think that is really hurting us a lot because all of a sudden, people who don't own any or are not close to NMR facility and don't have NMR instruments are interested to get things done. But what we can do is run samples ourselves, but we cannot get anybody to be physically there who hasn't used the facility before.
Well, thank you. Johannes, what were the measures you took at your university at Georgia Tech?
Okay, let me start my screen.
Let me
Karen.
stop sharing.
Can everybody see this now?
Not yet.
No,
Not yet, sir.
you may need to swap the screen if you have dual screen.
I have two screens. Okay, I'm not familiar with the software. So nobody sees my screen now?
No.
No, okay. What do I do now?
Well, we could move on with Mindley, and then you
Oh.
maybe have a few minutes to straighten out things. Does it work for you, Mindley?
Yeah, okay, good. Good. Sorry about that.
Yes.
No problem.
Let me see. I can share mine and see if it works.
You should be able to, yeah.
Okay. Can you see it?
Yes.
Okay, so yeah, I'm just showing some policy here in the University of Michigan Bio-AMR lab. So what we are actually doing to operate the AMR facility safely during the COVID-19 pandemic. So similar to other core AMR labs, we practice social distancing. And also we actually require gloves and face masks. And so gloves can cause some problems. So what we're actually requiring is that the users to put on a new pair of gloves when they enter the room, and after the use, just to remove the gloves. So that will maybe help a little bit with the problem with the gloves. And we provide hand sanitizer and wipes and so on. And so the users are required to clean those keyboards and wipe the mouse before and after use. So to keep the occupancy, so this in this AMR room, we actually have a iLab calendar. So users will reserve the instrument online, and they will come so that the instrument is one instrument per user. So that also helps. So finally, we have some very good experience with VNC. I think maybe everybody's using it. Users can just drop the sample and then remotely control the instrument. And it's also very useful for external users who cannot enter the building, and because we have some restriction on building access. So also we are using Zoom to share screen with users. If we are running experiments with them, they can just see how we are running, and also if they have problems with setting up parameters, we can work with them, and that helps them to get very good data. Yeah.
Okay.
Some experience. Yeah.
Well, thank you.
Yeah.
You have another slide, Mindley?
No, that's it. Yeah.
That's it. Okay, very good.
I'll stop sharing.
Yeah. Johannes, you want to give another try?
Now let's see. How's this now? Is the screen shared now? Okay, click share.
Ah,
There we go.
Yeah.
Okay, file to detect your microphone. Okay, check my. Okay, sorry. Can you hear me now?
Yeah, we
Yeah,
can.
we can hear you.
Good.
Go ahead.
So, my apologies again. So we have kind of a laboratory with 9 instruments, which is mostly a walk-in operation. We try with 2 managers. We try to keep it as much hands-on as possible. On this unfortunate Friday the 13th in March, all students were sent home. This is just for the spring break, and research was shut down the following Thursday. We were out of research. Only COVID-related research was allowed, which was kind of 2 NMR spectra were run between June and until end of June. And then there was the research ramp up, and up to now, 50% of the scientists are allowed to be on campus. Which means for the NMR lab that for some reason we are pretty much at full capacity. Some people work at night, but they all come to use the NMR lab. One of the things I think we are fortunate enough that Georgia Tech quickly established kind of a culture of COVID awareness, and this is throughout the entire campus. I like this signage. I like the culturally diverse, but everybody is wearing their mask. And we also have a voluntary weekly testing. It's not enforced, but everybody is kind of asked to be tested on a testing site in Georgia Tech campus weekly. So working with this, we established a routine. Everybody who comes in the laboratory has to wear a mask. There was a big discussion generally in the Department of Chemistry what type of masks, because if people use cloth masks, which they wear all day long, they might accumulate chemicals, not so much for the NMR lab, but for other laboratories. So the requirement is to wear the surgical paper mask. If you don't have one, we will provide it. Gloves, we ask people to wear gloves. We do it in that way that we provide one single-use gloves at the entrance. So if anybody is wearing gloves or not wearing gloves, you have to change gloves, change to a new pair of gloves as long as you're in the NMR lab. In addition, people have to disinfect their NMR workstations. We have a rather large lab, one lab which contains 8 of our instruments. There's sufficient space between individual workstations, so the rule is one person per instrument only. And in terms of keeping track who comes in, who uses the instruments, we have our own home institute-wide shared user management system where you sign up for instrument time, you log in, you register the time which you use the instrument, and this also keeps track who's using it. So there's no extra measures needed since everybody who comes in will log into an instrument and use it. So we have kind of a system where we track who is there. And generally, we have a good adherence. The one which has got the most problems and where we as the manager has to interfere is the one person per instrument. Students tend to be social, and actually science should be social. People should be talking, sitting on instrument discussion. That's the thing we just cannot have. So that's pretty much our scenario.
Very good. Marcus, how are things at Vanderbilt so far?
Well, I don't have any slides, so I just talk. It's been interesting. We actually did very similar things like Alex has kind of mentioned. We had pathways designed, we had the PPE all designed. And what I have to say, what hasn't been mentioned here, but at Vanderbilt, the administration has actually given us a lot of assistance. I was really impressed. We had individualized protocols we had to write and then submit, which really meant that we could write the protocols that would fit the facility. And it was not a one for everybody. And people in the administration basically read through each of these protocols and approved them accordingly. So that was really kind of outstanding to me. Obviously, we do have all the PPEs. The other thing that hasn't been really mentioned, what we did is initially we had a couple of users that had long-term samples, and we basically, in some cases, I set them up and ran them and provided them just the results for the users. In other cases, I set up the user to do remote login. So basically, they brought the sample, I took it, put it into the magnet, and then established a remote session so they could basically run their long-term samples. Okay. The small molecule facility was a little bit more impacted in that with the heavier traffic, they were really kind of closed for a little bit longer. Obviously, in our biomolecular facility, the long-term samples were running, but we were at lower capacity as well. And then eventually we were able to ramp it up and get basically things going again.
Well, good. We have a few more minutes left. Let's talk a little bit about training. The semester started, we have new graduate students coming in. These can be synthetic chemists, can be structural biologists. these students need to learn how to use NMR. That's part of their education, and it's a key mission at our universities. What is your plan, or what has worked already for you in terms of training? Who wants to go first?
I can say something.
Sure.
What has worked for us so far was that we have gone through logistics through Zoom meetings first, and then we have organized one-to-one.
The Zoom meetings were with multiple students at the time, or was it with?
No, one-to-one.
Okay, all right.
So that, because, you know, it was before everything opened, so we had time to do this. And that way, I was sure that the person was ready to get to instrument. So, but we didn't have that many people yet. So now it is a little bit tricky because our university still doesn't allow new users. So we are trying to develop the strategy, and I'm really eager to hear what the other experiences are, how to work on one-on-one and keep everybody safe. Because the shields and masks, they are not, believe me, they are not that comfortable. I can last half an hour max, and I think I'll suffocate.
I mean, one of the things we discussed at least was to develop some videos, but this is pretty time consuming. You know, just an introduction to how to operate an NMR spectrometer using standard operating procedures. Is it something some of you are also considering? I mean, is it something that could actually work together? Or what is your take on that? Maybe Johannes.
Oh, I just lost him.
Okay, just left. Yeah, Johannes is back. Yeah.
Sorry, my camera seemed okay. Sorry. So could I put the screen back on again? Huh? So what works with us is this is fine now. Sorry, share. So what works with us is that we thought about videos, but we really like to do it interactive, one-on-one. We have pretty much two feeds. One is we have a remote access to the NMR acquisition software, to TopSpin. We use AnyDesk. You could use any other like team viewers also as well. And we have a video conference. We don't use Zoom. We use BlueJeans. That's why I'm so unfamiliar with Zoom. But pretty much two feeds. We ask students, bring your laptop, and this works rather well. You had, instead of me sitting next to the student, the computer sits next to the student. I don't sit too far away behind in my office. Certain steps the student needs to put, insert this, to show him how to put the sample in a spinner, or to insert the sample insert an MIS. This one I do in person. I come with my mask. At least this sort of minimizes the contact, and so far the experience is not really horrible with this approach. It works well.
Mindley, what are you doing? Or what are you planning to do for training?
Yes, so at the first, I think from March to June, we are not allowed to do one-on-one training. So we don't have new users for that period of time. But then starting July, the university allow us to do training. So I'm still doing one-on-one training for students to show them how to load samples, how to run the spectrometer. But I try to reduce that time to like 15 to 20 minutes. And then for the following processing, data processing analysis, and also consultation, we're doing through Zoom. But still one-to-one. So far it has been working fine. So we haven't done any video yet, but if the students have some questions on some other problem, I prefer to write some step-by-step instruction and took pictures, so I can share with other users if there's problem again. Yeah.
Okay. Well, I understand videos is nice, but it's time consuming. Maybe that's something we could talk with Broker about. Clemens Anklin is here. Therefore, maybe it's an opportunity to develop something, not just for the centers participating at this gateway meeting, but actually for the NMR community worldwide.
It's worth mentioning Woong-Hee Lee in the chat just mentioned that NMR FAM has a bunch of workshop videos available for all levels of training new users.
Okay. I mean, the one of the challenges is obviously, you know, to agree on SOPs, that every facility has its own specialties, the own way of doing things. They want to have users really follow those. How do we can get some agreement? But I think it should be possible. You know, I think the benefits would be significant for everybody.
Yeah, we have this is Clemens. We have it on the books. Right now, we're still only partially reopened. So we can't even get the people in to produce such things.
Okay, no, I understand, but it's you know, if it takes 6 months or a year for individual centers, you know, maybe the pandemic is over by then. You know, therefore, it would be good to put something together within a month or so that actually our new students can really benefit, because there's a risk that they feel to be left behind. You know, they can hardly do rotations in the labs because of social distancing. They cannot really get training on the NMRs right now. Therefore, they take the coursework, but actually it's almost as if they hadn't joined the program yet. Therefore, that's something to consider. But Clemens, if you could update us on the progress with your video guys, then maybe we can provide some input as well. That would be really good.
Sure. Yep.
Yeah.
Good. Yeah, Marcus.
May I mention something? We actually have kind of resumed our regular training again, just modified. And that seems to be working. So what we do is we usually have a training session in the classroom in the morning where we go over things, and then in the afternoon we do training on the instrument. So we have smaller groups in the afternoon anyhow. Now with this modified thing, we just do the morning session over Zoom, and then the afternoon session, we spread it out a little more and do one-on-one over Zoom or with remote logins. And that, or even to some extent, one-on-one in person. So that has actually worked pretty well. And I think students actually do feel like they can get with the program and get the necessary tools to get going.
Very good. Okay, we're already a bit over time. I think we can finish this first panel. Thank you again, all the panelists. Really appreciate it. And we just do a short break to swap the panel. So our second part here of this panel will focus also on the same theme here, NMR challenges and opportunities during and post-COVID-19. But it doesn't focus on COVID itself, but more about the NMR opportunities and challenges in a rapidly changing environment in terms of other methods are becoming available. And when I started, you know, a few decades ago in NMR, the world of biophysics was relatively simple. There was NMR and then there was X-ray crystallography kind of as the gold standard in many ways. But over the past few decades, things have really become much richer and more complicated, but also more powerful in many ways. Therefore, we have all these other techniques that have really come out of age, have come of age, such as SAXS, FRED, single molecule spectroscopy, infrared in 2D, EPR, ESR, atomic force, mass spectrometry, and also in silico approaches, embedded computer simulations with modeling and docking approaches. And of course, most recently, cryo-EM has really come along and is taking out a lot of the oxygen, both in terms of the funding agencies as well as the high impact journals. And the question is here, so what is the role that NMR can and should play in such a changing environment, in such a rich environment? And even more so, they have, just over the past couple of decades, completely new branches of science have evolved, such as systems biology, that looks at complex systems in a completely different ways than the traditional structural biology and biophysics approach or the nanoscience as a new field looking at these intermediate small length scales. And there are synergies between all these approaches. Therefore, how do we make NMR useful? How do we keep it useful and powerful in such an environment? That's kind of the main question I would like to ask our panelists. And I'm really grateful that these 5 panelists have agreed to talk today. Angela Groneborn from the University of Pittsburgh, Sharon Campbell from University of North Carolina, Chapel Hill, Richard Krivacki from St. Jude Children's Hospital, Walter Chasin from Vanderbilt University, and Nick Fitzke from Mississippi State University. Therefore, I would like to ask Angela first. You have been working in this integrative structural biology field for a while. What is your experience? Where is NMR really useful? What are the opportunities that NMR can and should fulfill?
And if you allow me to share screen, if you go out of share screen, I'll try to put up a few slides.
Absolutely.
I have no idea whether you can see something.
Yeah.
Yes.
Just expand the
Yeah, hang on.
Then it's fine.
Need to get to my bottom. Okay.
Yeah.
So I think what we've all done over the last, I think now a decade, we have learned to work with colleagues in adjacent fields and methodologies. And although NMR, to the largest degree, unless it's imaging, belongs to what is called a structural technique, there are lots of other methodologies that if you want to solve a biological problem and ask questions, you have to combine and integrate the results that come from these different techniques. So mass spectrometry, cell biology, biochemistry, other type of spectroscopies, in vivo studies, and also computation, as was pointed out. Because in the end, we really want to see what are the details in the systems we are looking at that are related to physiological function. And as was pointed out, if you want to have atomic structures, there are basically only a handful of techniques: single crystal X-ray crystallography, solution NMR for solution, proteins you can dissolve, cryo-EM, you have frozen specimens, aggregates, solid state NMR, a lot of the structures that come from amyloid fibers, they are done by solid state. And indeed, ideally, even those techniques we want to combine. And I think NMR is very, very powerful in doing initial assessment when you're looking at a system. Like, for instance, in my group and in the HIV center that I direct here, we make all of the proteins labeled and do what we call quality assessment by NMR. And initially people were sort of not, a real spectroscopists were not happy about this. They thought, oh, we are now just being relegated to screeners, but it is really very powerful, because once we know we get good spectra, we can also set up drops, and then maybe sometimes things crystallize more easily. And I want to give just one example where solid state and solution NMR is very, very powerful. This also comes from our work here associated with the Pittsburgh Center for HIV Protein Interactions. We've been developing fluorine NMR now for quite some time. And in order to get assignments, we obviously have to do mutagenesis to get the amino acids assigned, which you can do very, very easily and quickly by solution NMR. But in the end, when you want to look at a very big system like the HIV capsid, this you do by solid state NMR. But if you would have to do that, all the initial screening, the mutants, which ones are possible to even look at, and we had to make a set of mutants that it's never easily predicted which type of substitution you can do and not disturb the structure. So we make all kinds of mutants. We check by this traditional HSQC or other solution spectra, and then we get the protein, the fluorine atom assigned to a particular portion, and then we can do the solid state NMR. And that allows us for the fluorine to, by correlation spectroscopy, now to get to long distances in very, very big systems, which by solution we couldn't do.
Okay.
The other example is obviously the overall structure of a native HIV capsid. There is no single methodology that could have given you that structure. The individual structures of the domains of the protein were done either by crystallography. The N-terminal domain was done by crystallography. The C-terminal dimer domain was done by NMR, and it was really only the NMR structure that allowed us to put things together, because sometimes when you have multimeric proteins, the arrangement of the monomeric unit in the multimer gets affected by crystal forces depending on which crystal lattice the protein crystallized in. And there were several crystal structures around, and it was impossible to put the system together with those crystal structures because the arrangement of the dimer interface was not the way as it was in the assembled capsid, the very long one. And for this one, we then had to do, I'm trying to get my laser color here so I can show something on the screen, probably not working. We had to do computation because we had to then put all the system together to first make the assembled structure in the test tube and to then convert what we saw there to the overall capsid, where we had to do cryo-ET on the capsid core. And the computation was absolutely essential to put all of those different bits together, because without the computation, we wouldn't have been able to do this. And then you have to, again, test all the system by mutagenesis, because when you do structural work, these structures are a particular representation of what goes on, but what you want to know is that what you see in the structures is what happens in the in vivo situation. And with the HIV, we can make those mutants and then show that what we see in terms of interfaces in our assembled overall capsid is actually important in the in vivo situation, because when we mutate those residues, the virus is no longer viable. And so I think the NMR can play a very substantial role in sort of being the glue between all of those structural techniques, and at the beginning get you to a situation where you can sort out how much you want to do with the different techniques. And I think there is really a bright future for NMR to be this joining link between lots of the other technologies. Because if you think about the cryo-EM, there will probably not a lot of structures that come to atomic resolution. People always call it sort of near atomic resolution. And if we can do individual domain structures by NMR and then check that the overall system fits well together, I think this is something where they will have a lot of work to do in the future.
A lot of good insights, Angela. Let's move on. Walter, do you have an example for the audience?
Yes. So I wanted to see if I can pop this up here. You see my screen now. Hopefully you see a slide.
Yeah.
That true?
Yeah, it looks good.
Okay. So I wanted to point out just a couple of things following up on what Angela said. Obviously, NMR fits in as an integrated technology, but I want to return you back to the idea, two ideas really. One is, never forget that NMR is the most powerful spectroscopic tool that we have available because of its ability to probe in an atom-specific way structure and dynamics. And that's essential to understanding function in this very detailed way. And I think the other point I want to raise is NMR has a role to play, in particular, when there are weak interactions. This is one of the few approaches beyond very basic biochemical approaches that can be used to look at very weak interactions. And weak interactions are often critical to biological function. And I don't have time to explain the details behind what I'm going to talk about. I just want to point out how we use it. But that's what we're looking at here is a very complicated machinery that's involved for repairing damage to DNA, the nucleotide excision repair complex. But we're going to focus just on one little piece here, which is shown in the circle, and that is the interaction between two domains. There's a primary interaction between these two proteins, XPA and RPA, that is much tighter. But the critical interaction happens right here on the substrate, on the DNA substrate, and it involves this XPA and RPA molecule. So this is an example where we need to get structural information, and it's an integrative approach involving starting structures, identification of interfaces, docking and refining. We use crystal structures, homology models, NMR, haddock, SAXS, and energy minimization to come up with a final structure. And so what we're looking at is the RPA70 tandem DNA binding domains and the XPA DNA binding domain, which binds at this interface between single-strand and double-strand DNA. RPA is a single-strand DNA binding protein, and these are just two of the critical interaction domains here. But in order to generate a structure of this model nucleotide excision repair corner, if you will, we started with a crystal structure of this RPA70AB bound to DNA. And then we had to build some sort of model to get a starting structure for the XPA DNA binding domain because there were two structures available, one in complex with some DNA and the other one an older NMR structure, both of which only contained this globular core, but didn't contain the full DNA binding domain. So we used this, a series of different modeling efforts, and came up with this starting model for the XPA DNA binding domain bound to this substrate here. That's what we're showing here. And in fact, the the RPA70AB fits on over in this region over here. So critical, NMR is the optimal approach for examining weak interactions, and in this case, for measuring the interfaces between the two domains, the XPA DNA binding domain and these two DNA binding domains of RPA70, but those two domains interact with each other, and that's what's shown here. Not going to go into the details. Everyone should know HSQC or TROSY HSMQC experiments that were taken on one side of the complex alone and then in complex with the other, black and red overlays. Same on the other side. We can use and look at the differences in the spectra to map out where binding occurs. We map that onto the structure. We use that to guide docking. In haddock in this case, we actually also use a Rosetta docking approach. And then we fit our intermediate model to SAXS data. And in this case, I point out this very high Perot exponent, which says that we could do reliable modeling in this case and come up with this final structure. And I'm just going to end by putting a plug for the PDB Dev, which is a new database dedicated to integrative structurals. And so you could take all of your data and, of course, your coordinates and deposit them in this new database.
Great. Just as a quick follow-up, what percentage of the total workflow included NMR, just as an estimate?
So we solved all the big problems using NMR and size exclusion chromatography, just finding out an optimal way to put everything together and the optimal DNA substrate. And the SAXS was, you know,
very good. Okay.
Very quickly after that. But NMR was instrumental and, of course, critical for those interfaces.
Okay, very good. Sharon, what is your experience in integrating techniques?
Yeah, if I can share my screen 1 second. Okay. So I'll be talking about a different project tomorrow, which uses highly integrative NMR techniques. So I've just included one slide here to give you an overview. Can you see it?
Yes.
Okay. So a major focus of our lab is the RAS oncoprotein. Mutations in RAS are found in about 30% of all human cancers. And an enigma in the field is that different RAS isoforms are found, or different mutations are found in different types of cancers. So for instance, you'll find a particular mutant of KRAS in pancreatic, colon, or lung cancer, but for NRAS, you tend to find them in melanomas and leukemia. So what I just want to point out some very interesting NMR data that pushed us in a completely new direction. So as Walter mentioned, NMR can provide site-specific information as well as information about dynamics. One point I'd like to pick up here is that if there are multiple populations that are detectable by NMR, you can also find them. So let me see if I can get my screen around. Sorry, let me get in screen mode here. Okay, so the first spectrum to the left here is a proton NMR HSQC spectrum of wild-type RAS, NRAS in red, in a mutant form of NRAS in blue. And what was surprising to us when we first took this spectrum is that compared to the wild type, we saw about 50 or 60 extra peaks. And we were able to detect peaks in regions that are dynamic in the wild type protein. So clearly this mutant adopted a conformation that changed dynamics. The other thing that was very striking is that if you take the same mutation in two different isoforms that are 95% identical to each other, what you find is what looks to be a major population in one isoform is a minor in the other. I'm just highlighting this here. So this just qualitative NMR data was a starting point for a whole new project, because what it indicated to us is that the mutants were different from the wild type, strikingly different, and that the same mutation in two different isoforms had different conformational ensembles. So what we did is we pursued molecular dynamic simulations to get an idea of what conformers were likely populated. We conducted cell biology experiments, biochemical experiments, and sure enough, without going into too much detail here, the conformers that we believe to be populated differently in these two different isoforms give rise to different biochemical properties in tumorigenic signatures. They also have different ligand binding pockets that could pave the way for mutation-specific drug discovery efforts. So my point here is this simple NMR spectrum, this signature was so distinctly different from the wild type, and then the same mutant in two different isoforms pointed us in a completely new direction. And I don't think anyone in the field of RAS would have predicted that the same mutation in two different isoforms would give rise to different conformational ensembles. These proteins, again, are 95% identical. So I just point this example out to give the power of just even qualitative use of NMR to provide new insights into protein structure and dynamics.
Okay, thank you. Richard, you work in structural biology and also in the emerging field of membraneless organelles. How do you harness the power of NMR in relationship with other techniques?
Thank you, Raphael, and hello, everybody. Sorry I came on this panel a bit late, but it was a busy afternoon. Yeah, as probably many of you know, we've become involved in studies of membraneless organelles and phase separation in recent years, and we've found NMR to be a particularly powerful tool for studying the flexible regions of proteins that undergo phase separation. So, I mean, some of the proteins that we study that undergo phase separation have both folded domains as well as disordered regions. And often the folded domains undergo oligomerization and create multivalency of the other domains, but it's very often the disordered regions that are mediating the weak transient multivalent interactions that drive phase separation. So NMR is particularly powerful in probing those flexible regions of proteins, even in the condensed phase. And increasingly we use complementary methods to gain sort of complementary insights. So we use SANS a lot to understand sort of the molecular spacing of molecules within condensates. And we use single molecule fluorescence to understand sort of the range of structural and dynamic features that exist for molecules in phase separated condensates. Since NMR gives you an ensemble average perspective, we find that single molecule fluorescence gives us, sort of complementary information about sort of the range of structural and dynamic features. And so I can share my screen and give you insight into one example, if I may. Here we go. All right, so I'll share this one. So can you see my screen?
Yeah.
Okay. Let's go to, oh, sorry, that's the wrong mode. There we go. Okay, so these are data for the nucleophosmin protein, which is a pentameric protein. So it has a pentamerization domain at its N-terminus. It has a 120 residue long intrinsically disordered region that has acidic tracks within the sequence. So there are 5 of those around this pentameric core. And then there's a C-terminal DNA and RNA binding domain. And so nucleophosmin is a nucleolar protein, and it phase separates with ribosomal proteins that have clusters of arginine residues that interact with these acidic tracks. And it also phase separates with ribosomal RNA through the multivalent RNA binding domains. And this slide pertains to interactions between phase separated nucleophosmin and a what's called a dipeptide repeat polypeptide. That is, in this case, 23 repeats of proline arginine. And this type of repeat is found expressed in certain cases of ALS. There's a gene repeat expansion that encodes dipeptide repeats, and several of them contain arginine residues, and these are very toxic to neurons and are causative in ALS in these patients. So this is a spectrum for this 164 kilodalton pentamer of nucleophosmin prior to interactions with a dipeptide repeat peptide. And then after, so these dipeptide repeats will initially phase separate with nucleophosmin, but at above a certain stoichiometry, so in this case 3 to 1, the droplets are disrupted, and the dipeptide repeat sort of causes the formation of megadalton structures that we've actually observed forming in cells when nucleoli are sort of intoxicated by this type of dipeptide repeat. So the green spectrum is a spectrum for nucleophosmin bound to 3 molar equivalents of this dipeptide repeat in the context of sort of megadalton assemblies. But we can see that resonances of the intrinsically disordered region are still detectable, as are resonances for the C-terminal RNA binding domain, which is not involved in interactions with the dipeptide repeat peptide. And so this just shows the extent of overlap between the two. So there are subtle chemical shift perturbations in the presence of the dipeptide repeat, and then these are indicators of where the toxic peptide is interacting with the residues in the IDR. And since the peptide has repeats of arginine, then those arginines are interacting with residues in the acidic tracts in the IDRs of nucleophosmin. And this just shows that the resonances of the CTD are not perturbed. And so, I mentioned earlier that we rely quite heavily on neutron scattering. So we did in this case. So I won't go into detail about this, but so this is just an image of the types of droplets that this DPR will form with nucleophosmin. But then when you add additional equivalents of the peptide, of the toxic peptide, then those droplets dissolve as illustrated in the upper little bubble. And when that occurs, then you see a striking change in the SANS curve. And so the features of these curves are indicative of a rod-like structure with the dimensions given in the upper right. And so by combining NMR data and SANS data, we've then come to a sort of low resolution structural model, but still a highly data informed model for what we think is going on. So here's the nucleophosmin pentamerization domain, and then here are the 5 IDRs. And what we think is going on is that the arginine-rich toxic peptides are insinuating themselves in between acidic tracts and causing them to cluster together into this sort of rod-like structure with the CTDs unengaged in this sort of interaction. And we think that a number of these sort of pentameric structures then cluster together to form the larger structures that that we characterized by SANS. And I should note also that we also have analytical ultracentrifugation data for these structures, which informed us about the approximately megadalton sort of size scale of these structures. So this is one example where we're combining NMR with SANS with analytical ultracentrifugation to provide insights into this sort of, it's not really a phase separated structure, but it's a structure that sort of formed through disruption of phase separated networks. So that's one thing I can share with the group in this realm of integrative structural biology methods.
Okay, Rich, your complex that you showed is more something like a dynamic nanoparticle than a traditional biomolecule.
Yeah,
Actually, our fifth panelist, Nick Fitzke, he's studying inorganic nanoparticles and how they interact with proteins. Therefore, how inorganic matter and biological matter do their interactions. What is your experience in working in such a cross-disciplinary field, Nick?
Yeah, thanks, Raphael. It's been fun. I think that's why we do all this, right? And I think that's one of the things that maybe we should say more of is that, you know, we do science because it's interesting and intrinsically valuable just to gain knowledge. Hopefully you can all see this at this point. Well, here, let me hit share. There we go. So yeah, we're interested in protein nanoparticle interactions. And so if it sort of touches on things that Angela and Walter and Richard and Sharon all mentioned, you know, they're often weak interactions. It's often difficult to crystallize. And so there's no one technique that's going to give you the answer that you want. And the reason we care about this is because if you're designing a nanoparticle-based drug, or if you want to make a nanoparticle do something, you really have to understand what's going on the surface. And so there have been lots of drugs that have almost come to market, but then they put them into a different type of environment, and now the endogenous proteins in the host are interfering with that nanoparticle function somehow. And this formation is called a biocorona. And in some types of inorganic molecules or inorganic nanoparticles, it's very, very strong. And so in our experience, gold is, you know, the protein binds extremely tightly so that the exchange rate is very, very slow, hours or longer. On other types of systems like the one Raphael works on, it's a much faster type of exchange process. And you can use that to get information on relaxation, which is pretty nifty. And so we get to use all of these sort of interesting relaxation sort of properties of materials as they exchange. But for most of our systems that we've studied so far, it's actually kind of boring. The protein kind of goes onto the surface and sticks. And so that's what we're showing here. It's just, you see the intensity go away. But because of that, it actually brings in this one of these, you know, to touch on the theme of integrative approaches, it brings in the ability to do all sorts of chemical labeling and chemical modifications of the systems that you're doing. And so, you know, I'm not going to go belabor this too much, but, you know, one of the earliest things that we tried doing was just, you know, we wanted to get some information on kinetics, and we only had access to 1D NMR, so why not, you know, use some filtering experiments to filter out the N15 labeled proteins from the C13 labeled proteins. And again, not terribly exciting or hot NMR, but very basic, uses very basic chemical principles, and now we can look at both proteins simultaneously just using 1D. Similarly, we've done some labeling and hydrogen exchange to monitor what's happening on surfaces, because again, the hydrogen, you know, once the protein binds this, you know, 15 nanometer nanoparticle, all of the NMR relaxes pretty much instantaneously. But you know, the protons and the deuterons that are in the backbone, you can actually trace that information if the protein comes off. And so again, using sort of chemical approaches, and then very recently we've started doing more sort of omics. So we've done some limited proteolysis and mass spec approaches to understand what's happening to these proteins in the presence and the absence of nanoparticles. And so again, this is one of those areas where you could say, well, cryo-EM probably isn't going to get us a whole lot of distance here. There have been some interesting solids NMR studies, but the mass of these nanoparticles, I understand, actually makes it a little bit challenging to do some of the spinning that you might like to do. And so, you know, we're often looking at other types of chemists in our department saying, hey, what chemical information can you tell me about this protein when it's absorbed, so that we can understand the physical principles of how a protein interacts with the surface and what it's doing once it gets there. And so that's really sort of all I have to say. And maybe if there's some time, we can entertain some questions from the audience generally. But otherwise, we can all go have dinner.
Okay. Well, thank you very much, Nick. I would like to just move on to or mention one other thing that is really coming along. And let me share again my screen here, and that's ultra-high field NMR. And this is actually in the back here, the first 1.2 gigahertz NMR at Bruker Bio has been back in November. When we visited, and this is Chris from the left and Alex on the right, when we visited Fellenden at the time when traveling internationally was considered as normal and indispensable for scientists. The grant that we obtained last year from the NSF to actually have a 1.2 gigahertz is coming along. We are renovating our space. This is the same space that Alex showed for the COVID measures, but we have now cleaned things up here, and we actually have a nice space here to accommodate the 1.2 gigahertz here with the 20-foot ceiling. And here's an instrument, the 1.2 gigahertz that has been installed, the second one delivered by Bruker at ETH Zurich, and it shows the size of these systems. And also here a few pointers here about the capabilities and the technology that are involved in making something like this happen. But I should mention that actually the very first ultra-high field NMR at 1.1 gigahertz has been delivered to the US, to St. Jude. And Richard, if you can tell us a little bit, because you're one of the very few with your colleagues at St. Jude who actually have hands-on experience with ultra-high field. Ours is expected to be commissioned maybe in 2022, maybe 2023. Therefore, we have to wait a little bit, although we're getting ready, maybe for an early delivery. But you have actually experience with such ultra-high field NMR. Can you share a little bit of your experience with us?
Certainly, Raphael. Thank you for the opportunity. And again, probably this is best done with by sharing my screen. So here we go. So this is So this is the 1.1 that's at St. Jude. It's in a so we now have 2 NMR labs, and so it is the flagship instrument, obviously, in this new laboratory. And so it depends on what people are interested in knowing about the instrument, but so I've queued up just a couple of comparison spectra just to give you some feel for what 1.1 spectra look like, in this case in comparison with 800 megahertz spectra. So these spectra on the left are the same that I illustrated a moment ago when talking about our integrative studies of the nucleophosmin DPR arginine-rich peptide complex. And so the in this case on the left is the red is the APO NPM, and the green is the complex, the megadalton complex. And so on the right are the spectra at 1.1 for the same 2 samples. And so you can clearly see, especially in the center of the spectrum, the benefits of enhanced dispersion, improved resolution in this region. And so, you know, as a consequence of this improved resolution, we can interpret chemical shift perturbations between the two states of the system to a greater extent at the higher field. And in fact, if you quickly compare left to right, you see that there are additional resonances present in the 1.1 spectrum and not in the 800 megahertz spectrum. So we don't fully understand the basis for that, but certainly suggests that maybe relaxation properties are changing, causing resonances to appear at 1.1 that don't appear at 800 megahertz. So that's one example. And another example, this is from Tanya Mittag's work by her postdoc Ivan Perrin. So here they're comparing the same sample at 800 megahertz and 1.1 gigahertz. So this is the dense phase of a phase-separated disordered protein, HNRNPA1. It's the low complexity domain from that protein, which phase separates under these solution conditions. I don't know the exact. So Ivan has prepared a large volume of these phase-separated droplets, and then has centrifuged them into one giant droplet and transferred that to probably a Shigemi NMR tube. And so he's shown sort of blow-ups of a few regions of the spectrum. So remarkably, even in the phase-separated state, where the protein concentration in the droplets is in the low millimolar regime, the polypeptide remains highly dynamic so as to give these quite sharp resonances. They're a little bit broader than in free solution, but still they're quite sharp. And so by comparing left to right, you can clearly see the advantage of having the improved dispersion associated with the 1.1 versus the 800. So these two examples really highlight the benefits of the higher field for improving resolution in spectra of quite challenging systems. I'll scan ahead. I don't want to go through these highlight reel things. I just want to get to this slide just to give people a little bit more information about the system. So our system reached field in just about a year ago. It was charged for about 4 weeks, I think. It did not quench, fortunately. So far, the drift and helium boil-off have been very stable, and of course, the drift was varying. Well, it was very high at first, and then it settled over a period of months to reasonable values, and it is now sort of well within Brooker's specification. And helium boil-off is quite good. So the magnet is very well behaved and stable. So we have two probes, 5 millimeter proton carbon dual resonance cryoprobe. And so this probe is used primarily for 3 and 4D proton carbon nosy spectra of methyl labeled or aromatic labeled large protein samples. Sorry for the typo there. And in addition, we have a 3 millimeter triple resonance TCI cryoprobe. So that's inner proton coil. And this probe is used, since Tanya and I are using the spectrometer primarily for disordered protein samples, many of which are phase separated. So maybe I'll stop there, and I'll be happy to answer any questions if there are any. I hope that gives you a little bit of insight into our experience so far with the instrument.
Well, thank you very much, Rich. Once ours arrive, we will certainly touch base with you to get more of your experience shared with us. Are there questions from the audience? I don't see much in the let's say, the Q&A. So maybe for Richard, Cassin Hall asks, the relaxation pathways at 1.2 gigahertz could confuse interpretation of relaxation data. What do you think will be the most difficult consequence?
We haven't done very many relaxation. We haven't done any relaxation studies at 1.1 ourselves. I mean, certainly one needs to take into consideration the effective field on T1, which then, you know, one needs to be mindful of delays between scans with longer T1s at higher field. And, you know, obviously a lot of relaxation parameters will be changing at the higher field. But
in terms of CPMG, Richard, I mean, the chemical shift dispersion picks up, therefore one should see more exchange effects, more minute exchange effects at higher fields. Therefore, that's in some cases an advantage. In other cases, one may not want to see those. But for those who are interested in this lower timescale dynamic, CPMG type of effects or SAXS effects, this actually is going to be an advantage.
yeah, certainly, you're increasing the you know the sort of spectral separation of the resonances for the different states. But you also need to be able to sort of keep pace with those changes with the pulsing frequency and with the and so it becomes challenging to be able to, you know, sort of apply sufficient RF power to be able to take advantage of, you know, those sort of changed sort of, I mean, the changing situation with CPMG type experiments at high field. And like I said, we really don't have very much experience yet with this instrument in applying those types of experiments. So that will have to come in the future.
I mean, the one thing I can see that at 15 CSA, for example, will be more important. Therefore, one needs a good grasp of what is the size of the CSA. Therefore, a good inclusion of that effect will be more important for quantitative interpretation in terms of order parameters and things like that. Therefore, there will be pros and cons, obviously, but I think the pros will clearly outweigh the cons at higher field, especially if one can combine some of that data also with lower field measurements.
Yeah, I think you'll have to, I mean, given the demand for the instrument, you'll want to do as much work at lower fields as possible, and then ask very specific questions when using the sort of ultra high field.
That's exactly the plan at our facility, that the current instruments, the 850s and so on, will serve as staging instruments, so that the time really can be saved for the special experiments when everything has been worked out at lower field for the ultra high field. Are there more questions? I don't see very much here. Therefore, people are getting hungry for dinner, I guess, and I think that's good. We have a full day again ahead tomorrow. And Marcus, let me know, is it 8:30 a.m. that the meeting starts?
It's 8:30 Central Daylight Time and 9:30 Eastern. So on the East, I guess you can sleep in a little bit longer.
Okay, very good. Well, let me thank all the panelists for an exciting panel discussion. I really appreciate it, and see you tomorrow.